Okay, so we are now recording and welcome to the last topic for the midterm period, which is enzymes. Okay, so enzymes are just special types of protein which has specific functions that are very vital to human life. Okay, and not only us, no, even animals, no, uh, and plants, they have enzymes. Okay, so ayun, these are just special kinds of proteins and they serve specific functions which allow us to live. Okay. So for this session, alamin natin kung ano yung enzymes, what are the classifications, what are the terms you have to know about enzymes, what is the active site of the enzyme, the enzyme kinetics, how it is controlled and regulated, and what are transition state analogs. Okay? So basically this term puro lang tayo keywords, okay? But I will give you some visual examples later on some specific enzymes para mas ma-recognize nyo na ito yung tinutukoy kong part ng molecule. Okay? So anyway. So with that, let's begin. Okay, so what are enzymes? No? So alam nyo, in your high school biology, that catalysts, no, or high school chemistry, that catalysts speed up the rate of chemical reaction. Okay? And in life, no, in living organisms such as tayo, instead of using catalysts that are used in chemistry, we use enzymes. Okay, so enzymes are the proteins that are used in enzymatic reactions, di ba? Kasi kapag yan ay inorganic chemistry reactions, usually ang enzyme diyan mga bakal-bakal eh. Ang gulat ka may bakal sa katawan mo, no? So hindi ganoon yung nangyayari sa tao. Instead, we use special types of proteins to do the enzyme catalytic functions, okay? So since it is a catalyst, okay, so it tends to speed up the rate of the reaction by a factor of 10 to 9 to 10 to 20 times, okay, over an catalyzed reaction. So suppose kunwari yung sugar, okay, will you say sugar spontaneously combust into CO2 and water? Hindi, 'di ba? Okay, unless sunugin mo 'yan. So the heat serves as the catalyst for your sugar to burn up. In our body, similar process happens pero walang apoy, okay? So, bale, kunwari, kumain ka ng asukal, what will happen? Okay? So, there are some enzymes in your body that will actually help break down those sugar molecules into smaller particles such as carbon dioxide and water. Okay? So, yun yung idea, no? So, hindi mo na kailangan ng apoy sa loob ng katawan mo. Ang kailangan mo na lang ay mga enzyme, no? That will help you break down your sugars, break down your glucose into smaller units such as carbon dioxide and water, which you will excrete afterwards. Okay? So, yun. Okay. So, pero kapag wala tayong mga catalyst, no, like fire or enzymes, hindi yan matutunaw. Hindi yan magiging carbon dioxide. Okay? So, yun yung purpose ng ating enzyme. They speed up the rate of the chemical reaction. And just as emphasized in the third bullet, without enzymes, life would be very impossible. Okay? Because typical chemical reactions involve high energy. So, when we say high energy, we are pertaining to reactions that produce or consumes thousands of kilojoules, okay? Which is equivalent to 1,000 degrees Celsius for others, di ba? Or 600 degrees Celsius. At that temperature, our proteins will denaturate. Masisira yung katawan natin. So imagine a world, no, wherein tayo, we don't have enzymes. Ano kaya itsura ng katawan natin, na? So, kasi, in order for the combustion reaction to occur, kailangan yung temperature mataas, no? Let's say 500 degrees Celsius, no? Para masunog talaga yung glucose. But thank God, hindi yan ang nangyayari. Okay? Because we are designed to have special proteins that can do similar tasks but on a milder condition nga lang. Okay? So, yan. And our cells contain many enzymes, no? Which serve different purposes. Okay? For each metabolic processes that we will discuss in the finals period, may makikilala kayo mga enzyme that is necessary for them to happen. Okay? So, yan. So, yan. So, ganun ka-important yung enzyme, ha? Kasi kapag walang enzyme, kailangan natin magsunog talaga sa buong katawan natin, literally. Okay? Susunugin mo lahat using flames. Pero, buti na lang walang ganun. Buti na lang may enzymes tayo. Okay? So, ayan. So, That's the reason why we are able to produce CO2 kahit mild lang yung temperature, 37 degrees, right? Yeah. So, ganun ka-important yung enzymes, okay? So, another thing with enzyme is that they are highly specific. When you say highly specific, ibig sabihin nun, sila ay may ina-attacking molecule lang na specific. Okay? So, sa dami ng molecule sa katawan natin, magtataka kayo bakit hindi sila lahat nagagamit, di ba? Okay? Ang dahilan nun is that our enzymes are specific kasi. If it is, let's say, glucose hexokinase na lang, okay? So, kunwari, yan ay hexokinase na enzyme. It will only attack glucose. Okay? So, that means other molecules in the body will be unaffected. Okay? So, ganun ka-specific yung enzyme. Pag ang gusto niya lang talaga kunin ay glucose, glucose lang kukunin niya. Kung gusto niya kunin yung S isomer, hindi niya kukunin yung R isomer. Okay? So it is highly specific in terms of the molecular structure and even in the stereochemistry. So ganun ka-specific yung enzyme. That's why our body, hindi siya nagkakagulo-gulo. Okay? So hindi siya jumbled mess. Rather, it is actually organized. No? Okay? So for specific reactions to happen, there will be specific enzymes necessary. Okay? So yun. So, ang tawag natin sa mga substances na kinukuha ni enzyme to do enzymatic reactions, we call them the substrate. Okay? So, yun. Yun yung panibagong term sa inyo, substrate. Again, so ano yung substrate? These are the molecules in which your enzymes react with, no? Para ma-produce yung ating products. Okay? So, yun. So, additional term, substrate. Okay? So, enzyme and the substrate. Okay? Or the molecule being reacted with. Okay? So now, bago tayo pumunta sa specifics ng enzyme, let's talk about the nomenclature first. Okay? So in the modern world, no, whenever you see ase as the suffix for the name of the protein, that is called enzyme. Okay? So basta may ase sa dulo, enzyme na tawag doon. Okay? So ganun na sa modern times. For example, hexokinase, phosphofructokinase. Okay? Ano pa ba? Uh, Isomerases. Okay, so kunwari, o yung mga yon, basta may ase sa dulo, meaning non-enzyme. Hulaan nyo yung ano, nasa laway natin. Ano tawag doon? Amylase, di ba? So, yung laway natin may amylase. Okay? Ase meaning enzyme. Okay? So, it is an enzyme that helps break down the sugar in our mouth no? To partially digest it. Okay? So, bali ganun yung idea natin. As long as it possesses the suffix ase, it means it can do enzymatic reaction. Okay? So, ito yung mga example, glucosidase, it can hydrolyze glucosides. Urease, it can hydrolyze urea. And lactate dehydrogenase, this is used to convert lactic acid to pyruvate. Okay? And vice versa. Ito yung dahilan bakit tayo nagmamuscle cramps. Okay? At ito rin yung dahilan bakit hindi tayo nalalasing. Okay? Kasi apparently in yeast, no, instead of them having lactate dehydrogenase, they have alcohol dehydrogenase. So, nangyari, when pyruvate is produced at the end of the glycolysis, nagiging alcohol yan. Okay? Ethanol, alak. Okay? Pero wala tayong ganun sa body natin. Mabuti na lang. Pero medyo sad din. Okay? Kasi, kung dahil wala tayong alcohol dehydrogenase sa ating katawan, lactate dehydrogenase meron, ano nangyayari? So when we do extraneous activities, activities that does na humihinto yung ating paghinga, nagpo-produce naman yung lactic acid which causes us to form muscle cramps. Okay? So yun dahilan bakit tayo may cramps. Okay? Ayan. Ako, pag nagsuswimming ako, madalas ako ma-muscle cramps. Siguro dahil payat ako. So mabilis lumalabas yung yung energy sa akin, mabilis lumabas yung heat. Siguro din kulang ako sa potassium, so I need to eat more banana. Okay? So yun yung mga factors no, bakit tayo pinupulikat etong molecule na to. Okay? So in humans, we have lactate dehydrogenase that converts pyruvate to lactic acid and vice versa. For yeast, they have alcohol dehydrogenase that causes pyruvate to be converted into alcohol. Ayan yung dahilan bakit tayo hindi lasing. Yung yeast, sila yung gumagawa ng alak. Okay? Anyway, going back, adenylyl cyclase. So this is one used in signal transduction. Okay, so one of the many steps in signal transduction. When you say signal transduction, ito yung cell signaling, okay, to induce specific tasks, metabolic processes. Okay, so one of the first step is to produce the cyclic AMP or cyclic adenosine monophosphate. Okay, so when CAMP is produced, signal transduction can occur. Okay? Ibig sabihin nun, it can signal our body to produce this hormone, produce this chemical reaction, so on and so forth. Okay? So for that to happen, we need this enzyme first, adenylyl cyclase. Okay? So basically, ang idea lang dito is that as long as they end in the A's, okay, they'll end at the A suffix. That means sila ay enzymes. However, there are some enzymes that doesn't end with the A's, especially those who were discovered before the modern period, okay, such as the pepsin and the trypsin. Both of them are enzymes used as digestive enzymes, no? So sila ay mga nakikita natin in our stomach and in our small intestines, no? So sila yung tumutulong to digest foods, no? To digest fats, okay? So yan. So although they don't end in the A suffix, okay, the reason behind that is that luma na kasi sila. They were discovered before the modern classifications, okay? So just for simplicity, ni-retain na lang, okay? But keep in mind that they are still enzymes, okay? So there are so many enzymes in the body, okay? So paano mo sila ngayon ika-classify based on how they perform, okay? So we have several types of enzymes. There are actually 6 of them, okay? So puntahan natin sila isa-isa. So first one, we have the oxidoreductases, okay? Oxido, ano kaya meaning yan? Oxidoreductases. Oxidoreduct, it should give you a hint that it can help us do redox reaction. O kaya inaaral nyo sa Anakem yung redox at alam ko iniiyakan nyo yung dati. Because redox reaction do happen inside the cells as well. Okay? So, in order for our body to do those reaction that will require redox reaction, hindi muna kailangan kuryentehin yung tao. Kasi sa Anakem, alam nyo, magpo-produce yun ng kuryente, di ba? But in the body, hindi ganun. Siyempre, gulat ka kung kukuryente ka na lang bigla, no? So, ang oxidoreductases, these are the enzymes, no, that facilitate the redox reaction happening in the body. So, for example, we have here the, okay, so we have here the lactate dehydrogenase. So, lactate is actually, this should be a carboxylic acid. So, hindi lang maganda yung pagkaka-drawing. So the carboxylic acid is an oxidized form of ketone. Okay, so this is a ketone. Okay, so basically, peroxide pala to. Okay, so basically, mangyayari lang dyan, hydrogens will be transferred to the NADH, no? So pag nangyari yon, redox na yan. Okay, so this molecule here will be reduced. Okay, kasi bababa yung charge niya. And this molecule here will be oxidized further to produce the pyruvate. Okay? So, ganun yung logic dito. Okay? So, may transfer ng electron, may transfer ng hydrogen to the other molecule, causing one molecule to be oxidized and one molecule to be reduced. Okay? So, in the lactate dehydrogenase, it is the lactate being oxidized to pyruvate, and NADH is being reduced to NADH. NAD plus is being reduced to NADH. Okay? Kasi bumaba yung kanyang charge. Okay? So, ganun lang. So, that's one of the many enzymes. Hindi naman kailangan kabisaduhin kung anong enzyme mismo yon. Pero yung mga examples dito, it will be helpful kung familiar kayo sa kanila. Kasi baka lumabas sa quiz. Okay? So, yung quiz kasi naka-test bank yun. So, I actually don't know what will appear sa part nyo. So, yun. Yung mga familiar enzymes na lang dito, yun yung i-familiarize nyo sarili nyo. Okay? So that's oxidoreductases. So instead of needing electrical voltage for this redox reaction to happen, ang kailangan na lang ng body is to use an enzyme, okay, to facilitate the transfer of protons, the transfer of electrons, okay, so that the redox reaction can occur inside the body without producing high voltage, okay. The next one, we have the transferases. The word itself should suggest na it transfers molecule. It can transfer specific parts of the molecules. Okay. Some examples of the transferases involves the transaminase and the kinase. Okay. So ano meron sa mga yan? Okay. So for example, we have here the transferase, seryl hydroxymethyl transferase. Okay. Ano kaya gagawin niya? E di magta-transfer ng specific portions of the molecule. Okay? So for this example, we have here the serine, okay? And then we have here the THF. Okay? So mangyari dan this CH2OH dito, pwede siya idugtong to the THF. Okay? So another example of transferase will be the uh, hexokinase. Okay? So kinases are one of the many examples of um, transferases. Let me show you. I have so many tabs here kasi ipapakita ko siya sa inyo mamaya. So hexokinase is one of the many enzymes used in glycolysis. Okay, and ano yung purpose niya? Its purpose is to transfer one phosphate group of the ATP to the glucose, so it becomes glucose 6 phosphate. Okay, so that entire phosphate group ito transfer niya kay glucose. And once we have G6P, then it is activated now. It could undergo either glycolysis, itong pababa, or the PPP. Ano meaning yan? The pentose phosphate pathway. Okay, so that's a different metabolic process. I think madidiscuss natin siya sa finals. No? Pero yun, basta before glycolysis even happens, the first step always is to produce G6P through the help of hexokinase. No? Kapag kinase, it transfers phosphates. Okay? Pero kapag transaminase, it transfer amines, okay? Amino groups, okay? Ayan na, tignan nyo, basta may kinase, may mga ATP, okay? Kinase, may ATP na naman, okay? Kinase, okay? Kinase, okay lang yan. Basta alam mo yung spelling, okay? So those are the transferases. They help transfer specific parts of your molecule in order to either activate them or to produce the final product, okay? So the next type of enzymes are hydrolases. Okay, so what's hydrolases? Hydrolases help in the cleavage of the bonds by hydrolysis. Okay, so ibig sabihin you add water para yan ay maputol. Okay, so some examples are ligases and lipases. Lipase, no? Okay, so ano ginagawa ng lipase? Pamputol ng lipids. Okay, so in order to cut the triglycerides, yan, gamit ka ng lipase, no? So what will happen there, oops, ba't ko na-exit? So what will happen here, so for example, we have this urease, okay? So this is an enzyme used to cut our urea, okay? So mangyayari dyan is that when water is added, yan, mapuputol yung amine bonds, so that will produce ammonia and CO2, okay? So apparently, no, may nabasa ako sa internet, especially about pusa. So ano meron sa pusa? Marami kasi akong pusa sa bahay. So pag umihi sila, alam mo na mapanghi talaga. May nabasa ako dun sa sa internet. Sabi pala, I'm not sure talaga, no, pero hindi ko na verify. Medyo na convinced ako na. Anyway, so ang idea dun pala is that minsan, no, kapag umihi yung pusa, then super duper yung amoy nun. What happens is that yung urea, it breaks down into CO2 and NH3. Okay, it will take days now for that to happen, okay, or few hours for that to happen. Okay, so bale yung ammonia, ito yung super masangsang na amoy. Okay, and yun, na pag umihi yung pusa, it will take few hours until ma-breakdown into yan ammonia. Okay, so sa body natin nangyayari din to, pero hindi naman hours yung kailangan intayan, okay? At hindi rin tayo mangangamoy pusa, okay? Kasi ang ginagawa ng ating body is that yung mga amine groups, nire-recycle na yan, nila yan to produce amino acids, okay? So, ibang metabolic process pa yan. Then, yung CO2, ina-exhale natin, okay? So, yun lang. Side kwento lang. And then, yung excess urea sa body natin, sometimes they are excreted sa urine. Kaya nga, urea, okay? So, yun. Another type of enzyme is the lyases. Ano meron sa lyases? It cleaves specific bonds. Okay? So, meron lang sa lyases is that may formation tayo ng double bond dito. Pag hydrolysis, pinuputol talaga yung bond with the help of water. Okay? So again, lyases and hydrolysis, they are kind of similar. They cut the bonds. However, for lyases, may formation tayo dito ng double bond. Okay? So for example, the pyruvate, okay? So if you use pyruvate the carboxylate, it will be converted into acetaldehyde and CO2. Saan napunta yung double bond dun, sir? Okay. So the lowest structure of CO2, dalawa yung double bonds niya sa magkabila ang gilid. While for this COO minus, isa lang yung double bond dyan. Okay. So to convert yung COO minus to carbon dioxide, yung double bond mafo-form between carbon and another oxygen. Okay? So, yan. You will have your CO2. Okay? And then isomerases, from the word itself, it helps in racemization of optical or geometric isomers. Okay? So basically, ito yung ginagamit if you want to convert a molecule into another isomer form. Okay? So, pwedeng R to S, S to R. Pwede rin naman from one constitutional isomer to the other. Okay? So, ganun yung ginagawa ng isomerases. Okay? So, just like in my previous example here, okay? So we have some isomerases in the glycolysis such as the TPI or the triose phosphate isomerase. Okay? So gagawin yan, mangyayari is that itong G3P na to, maka-convert yan into this. Okay? So yan. So mag-reshape lang siya. Okay? Even this mutase, I think mutase is another isomerase. Okay? If tama yung pagkakaalala ko. Okay. So, gagawin lang na itong mutase na to, para lang siyang isomerase. Yung phosphate, instead na nasa third carbon, ilalagay niya sa second carbon. Okay? So, ayun. So, anyway, more of that pagdating natin sa finals period. And then lastly, ligases. Okay, so ano meron sa ligases? They form bonds between carbon and oxygen, sulfur, nitrogen, coupled with hydrolysis of high-energy phosphates. Okay, so let's take a look at this example here. So we have here pyruvate, no? And if you want to convert it into oxaloacetate, which is one of the precursor in Krebs cycle, okay? So yan. So you need you need ligases here. Okay? So, in summary, 6 yung uri ng ating enzymes. Okay? Huwag nyo nakabasaduhin yung lahat, no? Ang mahalaga lang dito is yung classification niya. Okay? So, oxidoreductase, they help in redox reaction. Transferase, they help in transfer of specific portions of the molecule. Some of them are considered transaminase and some of them are kinases. Okay? Pag sinabing kinase, ano tina-transfer niya? Phosphate. Kapag transaminase, ang tina-transfer niya, amino group. Okay? And then we have hydrolase. Hydrolase are used to cut bonds. Okay? Pag pinutol yung bonds with water. Okay? Yun yung hydrolases. Okay? And then we have lyases. So ito naman, ikakat yung bond through formation of double bonds. Okay? Isomerase, these are used to create isomers, no? Whether that's stereoisomers or geometric isomers or constitutional isomers. Okay? And then ligases, ito naman yung ginagamit to cut the bonds. Okay, to form the bonds coupled with ATP reactions. Okay. So yan. So yun yung classification of our enzyme. And now, let's proceed back to the enzyme terminology. Okay. So again, yung topic natin, puro terms lang talaga to. Okay. But you have to see the interconnection of one term to the other. Okay. So now, alam na natin yung mga enzymes, yung mga uri niya. Now, let's talk about the enzyme molecule itself. Okay. So here are several terms that you will need to know in order to understand how enzymes work. Okay. So, first we have the APO enzymes. Okay. So, ano meron daw sa APO enzyme? These are inactivated forms of enzyme. Okay, without its non-protein uh, moiety. Okay, so ano lang meaning niyan? So APO enzyme is the inactivated form of enzyme. Okay, so in the sense, no, before yung enzyme maging enzyme talaga, it will be an APO enzyme first. Okay, so what's a good example of this? Okay, so pakitaan ko kayo ng good example of this. So this is the human insulin. Okay, so it is just 21 amino acid chain. Okay, so this is the activated form of insulin. And mapapansin mo, dalawang magkaibang strand yan ng protein sequence. Okay, pero before yan gumana, may precursor yan. Okay, may precursor yan. Ito yan. Ito ba yan? Sorry. Ito. The proinsulin. Actually, meron pang tinatawag na pre-proinsulin. Okay. So, ang mangyayari, so for the insulin to work, no, it should exist first as this one, the proinsulin. Okay. So, ano meron dito sa proinsulin? Let's take a look, no. As you can see, yung enzyme niya, yung protein chain niya ay interconnected lahat, no. So, dugtong-dugtong pa siya. Okay. Dugtong-dugtong siya. So, ito yung APO enzyme na tinatawag natin. So, it's just a protein. Sorry. May nakasabit sa lalamunan ko. Okay. So, it's just a protein, no? Then, when you cut it, no? Mamaya may nalaman natin ba't kailangan i-cut yan. So, when you cut it, no? It turns into two different strands of proteins. Okay? And this one is the insulin. And ito na yung ginagamit for regulation of sugar, especially glucose levels in the blood. Okay? So, yan. So, before it becomes a real enzyme, it first needs to be an APO enzyme muna. Okay? The inactive form of the enzyme. Okay? So, yun yung APO enzyme. Siya yung inactive form of the enzyme. Okay? And then we have the cofactor. So, ano yung cofactor? Sabi dito, this is a non-protein portion of the enzyme necessary for the catalytic function such as zinc and magnesium. So, I have example here. No? Asan ba yun? Ito ba yun? Ito. So, I have here a zinc. Okay? So, zinc is one of the cofactors necessary, okay, in the formation of zinc finger, which is, if you know, no, zinc finger is a one of the enzymes, no, one of the functions of the enzymes needed in the replication process. Okay, so it helps cut your DNA, di ba? Yung zinc finger, nakaganon yan, tapos puputulin niya yan. Okay, so yan, mapapansin nyo, so we have proteins here. Okay, so may mga proteins tayo that will surround our DNA, okay, ating double helix. And in order for that to be activated, you need to have an enzyme, you need to have a cofactor, which is the zinc, okay? So that's why zinc is very necessary in your diet, okay? Kaya mapapansin nyo, pag kayo may sakit, ang sabi ng doktor niya, minom ka ng may zinc, no, other than the vitamins, because apparently zinc, no, it helps produce healthier cells na. Okay. So kasi mas mabilis na yung DNA replication, okay? Ipmat na yan, no? Interphase, prophase, metaphase, anaphase, telophase, na yung bibilis ata with the help of zinc. Basta ang alam ko lang, zinc is necessary for cutting your DNA to reproduce, okay? So, ayan, kaya yung sinasabi sa atin ng doktor natin, okay? So, kapag may sakit, other than the vitamins, inom ka ng zinc because it can help, no, in some enzymatic functions, one of which is yung sa ating DNA, okay, para mabilis dumami ang healthy cells natin. Okay, so yan, so hindi siya obvious at the at the macro scale, but if you zoom in, doon mo makikita yung importance ng zinc. Okay, and as you can see here, okay, so it appears that our zinc, saan siya connected? So this is histidine. Okay. So this is histidine and this is cysteine. Okay, so cysteine and zinc. Okay, so probably yung cysteine dito nawala na siya ng H, no, because as you know, it is protonizable. Okay, so may pKa yung SH group. So yan, so yung charge na negative dito and yung lone pair ng nitrogen dito sa histidine, they complexed with the zinc. Okay, so yan, so pag nagkaroon ka ng zinc finger, ayan, matutulungan niya yung DNA replication process. Okay, so that's cofactor. So sila yung mga metals, okay, non-organic molecules that are necessary in our metabolic processes. Okay, so this is from a different PowerPoint. Let me just show you. So paki-screenshot na lang. So these are some cofactors needed for some enzymes. Okay, so yan. So for potassium, okay, ito ay ginagamit for the pyruvate kinase. Okay, magnesium is used also in hexokinase. Ribonucleotide reductase, manganese, alcohol dehydrogenase, kailangan din pala ng zinc doon. Glutathione peroxidase, kailangan niya ng selenium. Copper 2+ for cytochrome oxidase. Okay. Site C, ito yung nasa electron transport chain, and dinitrogenase, molybdenum. Okay, so please take a screenshot na lang because this is the PowerPoint I used before sa edu class ko. So, para na may additional idea kayo kung ano yung mga cofactors needed for some enzymes. Okay? So, let's go back. Asan na ako? Ito. So, balik tayo sa main PowerPoint natin. So, cofactors. Ayan. So, ayan. So, thank you, Rebecca. May picture na dyan. So, cofactors, yun yung mga metals that are needed in the formation of your matured enzyme. Okay. Ganun pala itsura ko sa screen nyo. And then, you have your coenzyme. Okay. So, coenzymes naman, these are organic molecules. These are non-protein component of your enzyme, okay, which also helps it to be activated. Okay. So, ano example natin dyan? Ito, paki-screenshot din from a different PowerPoint to. Ayan. So, yan. So, these are your coenzymes. And saan sila nakukuha? Basahin nyo yung dietary precursor. Vitamins. Okay? So, yan. So, that means you really need to have vitamins included in your diet. Okay? And you need some inorganic minerals, no? Such as zinc, magnesium. Okay? Para gumana yung ating mga enzymes sa body. Okay? Kasi kapag walang mga ganyan in our body, okay, our mahihirapan tayo mag-produce ng mag-produce ng mga enzymes na may specific enzymatic functions. Okay? So, ayan. Okay, balik na tayo. Na-screenshot nyo ba? Okay, so ayan, may screenshot. Okay? So, ayan. So, again, cofactors, these are metals, no, which are used to bind with our proteins in order for it to achieve a structure, no, that can help in its enzymatic function. And then, kapag coenzymes naman, these are organic molecules that are non-proteins that are necessary in enzymatic functions, and they are primarily used in diets. Okay, they can be primarily obtained from our diet, no. So ibig sabihin na to, kumain ng gulay kasi gulay is rich in magnesium. Okay, may zinc din dyan. Okay, and then uminom ng vitamins. Okay, so gulay pa rin. Okay, so yan, so you really need to incorporate gulay in your diet, no, so they are rich in those molecules and metals. Okay. Then substrate, just as I told you, substrates are the molecules na ginagamit ng enzyme, no, para magkaroon ng chemical reaction. So siya yung binabago ng enzymes natin to produce our product. Okay? Holo enzyme. So when you say holo enzyme, this is the entire enzyme when our cofactor or coenzymes are assembled in one units. Okay? So kapag yung cofactor or yung coenzyme idinikit mo kay enzyme, tawag yun holo enzyme. This is from a different PowerPoint again. Paki-screenshot na lang. Yan. So, yan. So, this is from a different PowerPoint. Paki-screenshot na lang. So, dito nyo makikita yung function ng ating cofactor and our apo enzymes. Okay? So, yan. So, bago siya gumana, kailangan niya ng cofactor or sometimes coenzymes. Okay? And pag nagsama silang dalawa, tawag doon, holo enzyme. Okay? Holo meaning isang buo. Okay? Whole. Okay? So, kailan ulit tayo nagkakaroon ng holo enzyme? When you have your precursor, apo enzymes, idinikit mo ang cofactor or coenzyme, you will have the entire functional enzyme called holo enzyme. Okay? So anyway, yun lang naman yung mga slides na gusto ko kunin sa PowerPoint kong isa. Okay? So yun. So and then lastly, you have your active site. Okay, so your active site is a special crevice or pocket in your enzyme. Kasi 'di ba last time nakita niyo yung mga enzymes na share ko screen ko. Yung enzymes natin, irregular yung shape niya, 'di ba? Tapos makikita niyo may mga depressions doon, may mga crest doon, 'di ba? May sinabi akong ganun. Yung crest na yon, that is actually the active site. Okay? Ano nangyayari sa active site? So in the active site, this is where your substrate can enter. So pag pumasok dyan yung substrate mo, dun mangyayari yung transferase, no? Isomerase, no? Dun mangyayari yung mga ganong reaction, no? So, dun niya babaguhin yan into a different product, pwede niya dugtungan yan ng molecule, pwede niya hatiin yan, so on and so forth. Okay? So saan nangyayari yon? Sa active site. Okay? So I'd like to share to you one enzyme here. Ito, napakita ko na to sa inyo, 1QHA. This is the human hexokinase type 1. This is the enzyme used in glycolysis in humans. Okay? So, makikita nyo, may special cleft. Okay? May crevice, may paloob na ganun sa enzyme na to. That is actually the active site. Ayan, kikita nyo yun. Parang may empty space sa loob, di ba? That's actually the active site. Okay? However, itong sa tab na to, this is a hexokinase from Schistosoma mansoni. I googled that. Yan ay ano pala, yung fluke. Yung mga nasa palayan. Okay. So that's fluke. Okay. So this is the hexokinase of fluke. Okay. And as you can see, it also has crevices. Okay. May mga depression siya. And for example, ito, look at this. This is alpha-D glucopyranose, which is sugar. Okay? So, nangyayari, doon sa active site na yon, doon sa mga depressions na yon, doon pumapasok yung ating substrates such as sugars. Pagpasok ni sugar doon, ayan, tignan nyo. Didikit siya sa mga other protein residues and doon na mangyayari yung chemical reactions. Okay? So that is the purpose of the active site. Okay? So again, you can see active sites in your protein 3D structure kapag may nakita kang mga palubog doon, no? May mga depressions doon. Yan ay tinatawag na active site and that is where your substrates enter. Okay? Such as, for this case, glucose kasi this is hexokinase. Okay? So pagpasok doon ng glucose, dudugtungan siya ng phosphate, then lalabas uli siya dyan. Then papasok na naman yung iba pang glucose and lalabas sila. So ganun yung ating enzymes. Okay? Yun. Okay, so those are the terms in enzymes. We also have what's called allosteric site. Okay? So other than the active site, meron ding allosteric site. No? So yun yung mga positions in our proteins na nakapalubog din pero hindi doon pumapasok yung substrate. Okay? So I'm not really sure where the allosteric site is here. Probably ito. Okay? But ayun yung active site. Okay? So, ito yung active site, yung pinakamalalim na crevice doon. So, doon papasok yung substrate mo. More later, tignan natin yung allosteric site in much detailed illustrations. Mamaya na lang. Okay? So for now, these are the enzyme terms we need to know, and each of them serves different functions. Okay? Okay, so APO enzyme, ito ay ginagamit, ito yung inactivated form of the enzyme. For you to activate it, you need to have cofactors and coenzymes. Cofactor, metals, and coenzymes, these are organic molecules derived from vitamins. Okay, and pag pinagsama mo yung dalawang yan, you will have your holo enzymes. Okay, and the substrate enters the active site. Okay, that is the cleft or the crevice wherein your molecules can bind and react. Okay, so again, here are the enzyme sidekicks, the cofactor and the coenzymes. Okay, so ngayon, punta tayo ngayon sa binding process ng substrate to the enzyme. Okay, so sabi ko, yung ating substrate, it goes into the active site, okay, wherein the enzyme catalysis will happen. Okay, so when your substrate enters the active site of your enzyme, what we have here is called the enzyme substrate complex. Okay, and so it is the enzyme substrate complex, and sometimes we need coenzymes. Okay, in order for your enzymes to work. Okay, the mechanism of your the mechanism of reaction ng enzyme and substrate mo may fall under these two models. No? We have the lock-in key and the induced fit models. Okay? So basically, in the lock-in key model, it states that the enzyme has a rigid shape. No? Ibig sabihin, fixed talaga yung shape niyan for a specific molecule. Okay? So for example, in this picture here, we see that yung shape ng enzyme is not changing at all. However, the more acceptable model of our enzyme is called the induced fit model. Okay. So ito, ang assumption lang natin dito is that yung enzyme rigid siya. So ibig sabihin one molecule will react with one enzyme only. However, we also have this more acceptable model called the induced fit model. So ano meron sa induced fit model? So it assumes that your enzyme is not really rigid at all. It is flexible. So that means it can accommodate different types of substrate then. Okay? And as you can see kanina sa previous examples natin, totoo yun. Okay? So totoo na yung ating mga enzymes. No? Hindi lang siya limited to one substrate. For example, the lactate dehydrogenase. Pwede siya gamitin ng pyruvate, pwede rin siya gamitin ng lactate, di ba? So, ibig sabihin, induced fit model is the best model in terms of the enzyme action. Okay? Sa lock and fee, isang susi para sa isang pintuan. But that's not the case for the enzyme because remember, it possesses electrostatic groups. No, yung ating protein, yan ay squishy yan. So that means it can really accommodate changes in the shape. Okay? As long as allowable yung shape na yun. Okay? So yan. So that's why induced fit model is the best model for enzyme action. Okay? And ayun na nga, sabi ko kanina, enzymes are highly specific, meaning they will interact with limited substrates, okay? With few options of substrates in performing the same type of chemical reaction. Okay? So hindi pwede ibang substrate na iba yung chemical reaction. So that's why we have different classifications of enzyme because all of them works at different purposes, okay? So we have different types of specificity for enzyme. We have the absolute specificity. So ibig sabihin, these are enzymes that are absolute, no? Ibig sabihin, iisa lang talaga yung kukunin yung mga substrate. Okay? So for example, catalase is just an enzyme used in hydrogen peroxide, nothing else. And then we have the group specificity. Okay, so pag sinabi natin group specificity, that means if they have the same functional group, then they can react. Okay? So for example nga, yung lactate natin and yung ating pyruvate, they both have carboxylic groups. So that means they can be both accommodated by our lactate dehydrogenase. Okay, so lactate dehydrogenase possesses that group specificity. Okay. Another thing is the carboxypeptidases. Okay, as long as you have your carboxyl, it can react. Okay. Linkage specificity. Okay, so ano naman sabi dito? So these are enzymes that react in only particular types of bonds, nothing else. Okay? So for example, pag sinabi phosphatases, it will only react with the phosphate linkages. Okay? Okay, so ganun. Stereochemical specificity. This is about the stereoisomerism naman. Okay, so if your sugar is D, it will not be accommodated in an L sugar accommodating enzyme. Okay? So, kunwari, you have your R amino acid, you have your S. So, hindi niya i-accommodate yung kabila. Okay? So, yan. So, yun yung ating enzyme specificity. No? Sometimes they are absolute, meaning they are restricted to one function only. However, sometimes if they have the similar functional group, they will accommodate it. If they have specific bonds present in the molecule, they can accommodate it. And then stereochemical specificity, this is about the 3D orientation of your molecules in space. Okay? So, ba't kaya nilagay yan? Anyway, so enzyme activity. So, dito na tayo. So, enzyme activity, just as I said, no, enzymes are used to catalyze the reaction. When we say catalyze, we speed up the reaction. Okay? So, we are familiar with this graph here. Okay? So, nakita nyo lang to nung chapter 0 natin. Yung alam ko nakaganon kay ano daw. Kasi ang daming thermodynamics kung sinabi. Okay. Pero you're familiar with this one. Okay? So, this is our free energy diagram in which we can see the energy of your reactant and the product. Without catalyst, you can see this great barrier here. No? Great barrier. Okay. Malaking energy barrier. This energy barrier is called the activation energy. Okay? This allows your reaction not to happen. Okay? Siya yung dahilan bakit hindi nangyayari yung mga chemical reaction. Because if there are no enough energy for your reactants to overpass uh, this activation energy, hindi siya mapupunta to the product side. Okay? Kaya nga yung sugar natin, pag inayaan mo lang yan nandyan sa paligid, hindi yan magiging carbon dioxide and water. Why? It has a great activation energy. So, pag hinayaan mo lang yung asukal mo na nandiyan nakatiwangwang, hindi yan magiging CO2 and water. However, when you heat it, providing energy and the form of heat, tataas yung ating energy ng reactant and then, yun, mapoproduce na yung products. In the body, of course, hindi natin pwede initin yung katawan natin. Otherwise, cremation tawag doon. Okay? So, instead, ang gagawin ng ating body, it will use enzymes. Okay? So, enzymes such as hexokinases, amylases, okay? Mga pepsin, trypsin, okay? So, yung mga yon ginagamit nila yon, okay? In order to provide alternative pathways, okay? So, again, enzymes provide alternative pathways, okay? So that the reaction will proceed at a minimal energy, okay? So, ganun. So, instead of requiring you to burn your sugars literally, it will burn your sugar on a different mechanism. Okay? Pag sinabi natin, di ba, burn your fats, hindi naman sinabi, apuyan mo talaga yon, no? You do something else to burn it on a different way. Okay? So, ganun. So, yun yung purpose ng enzyme. Okay? So, since our enzyme provides a different mode of mechanism for the chemical reaction to happen, ano mangyayari? Mas bababa yung activation energy. Okay? So, yan. May activation energy pa rin, yes, pero kaya na i-provide ng body yon. Okay? So, siguro kaya na yun at room temperature. No? So, that's why tayo at room temperature parati yung ating katawan, di ba? So, yun. Para ma-maintain niya yung enzymatic reaction sa atin. Okay? Okay, so enzyme activity can also be measured in terms of the speed. Okay? So, if we graph the speed of our enzyme activity, Okay? With respect to the enzyme concentration, substrate concentration, temperature, and pH, we can see similar patterns. We can see recognizable patterns. Okay? So, for example, so when you increase your enzyme concentration, the speed of reaction increases. Obviously. Bakit? So when you have many enzymes, no, pag marami kang enzyme masyado, ibig sabihin, yung substrate mo, mas mabilis sila ma-accommodate. No? So that's why the speed of the enzymatic reaction increases linearly with increase of enzyme concentration. Parang ano lang yan, no? So for example, voters registration is extended until October 30. So, pa-register na. So, kunwari, tignan nyo, kapag yung sa voter registration, dinamihan nila yung mga booth, no, wherein people can do biometrics there, edi mas mabilis yung process talaga, no. So, think of the booths, no, yung mga tao doon as the enzymes, no, and you are the substrate. You go to the enzyme, yung booth, and then after nun, registered ka na, lalabas ka na sa enzyme, mapapalitan na yun ng panibagong substrate. Okay? So, ibig sabihin, mas marami yung booth, mas maraming mapaprocess over time. However, let's take a look at this one. If you increase the substrate concentration, on the other hand, look at what happens to the graph. Okay? The graph seems to plateau. Okay? Meaning ng plateau, nagpa-flat. Okay? So, yan, plateau. Okay? So, yan. So, nagpa-flat siya at the end. Bakit? Kasi at this point, no, kapag super dami na ng substrate natin, yung enzyme mo limited. Ano mangyayari? Maging fixed na yung rate niyan, okay? So, ganun na nga. For example, in our COMELEC registration, maraming gusto bumoto. Bakit? Alam nyo na naman nangyari sa Pilipinas. Okay? So, sa sobrang dami ng gusto bumoto, nagdagsaan sila sa booth, no? Ang problema, for example, kunwari, sa isang polling center, 3 lang yung booth, kunwari. Okay? Tapos, libo-libo na yung tao na nakapila. So, sa tingin nyo, pag dinagdagan nyo pa yung mga nakapila, bibilis yung process. Hindi, di ba? So, yun yung nangyayari dito. Kumbaga, pag sobrang daming enzymes na, pag sobrang daming substrate na, may limit na yung enzymes na kaya niya i-process. Okay? May limit na. Hindi na niya kaya mag-produce ng mas maraming products. Okay? So, that's why the rate seems to flat out, because the enzyme feels saturated. Okay? Parang kayo lang din to kapag Hell Week, 'di ba? Pag sobrang dami na naming tinambak sa inyo na gawain, ano na kayo? Parang kahit anong puyat gawin mo, parang hindi ka na productive, 'di ba? Na-feel niyo 'yun, 'di ba? Parang ayan, nag-sacrifice ako isang araw para mag-work. Wala. Wala pa rin ako nagagawa. So, yun. Okay? So, substrate is your, mga quiz namin sa Canvas. Tas kayo yung enzyme. Ang dami na namin binigay sa inyo. Tas kayo, kunwari, mabagal kayo talaga gumawa. Mabagal din yung, process mo talaga. Okay? So, you will come to a point wherein you are getting saturated. Okay? And then, next one, we have the temperature. So obviously, as we can see here, the peak enzymatic reactions happen at somewhere here, no? So let's say 37 degrees Celsius, which is the normal body temperature. Okay? Why? Kasi if yung katawan natin wala sa normal body temperature, sometimes our proteins can get denaturated. Okay? For example, at higher temperature, nasa oven ka. Ano mangyayari? Sobrang init na, wherein your proteins can get denatured, and remember, enzymes are proteins, so they won't work at higher temperature. Kaya nakakatamad mag-aral kapag mainit. Isipin nyo, ganun na lang din sa enzymes na. Pag sobrang init na for the enzyme, masisira na siya. Okay? At lower temperature, on the other hand, okay? Parang ito naman kayo, no? Mag-aral nga kayo, pero nasa aircon, inantok naman kayo. Okay? Kasi nangyayari is that at lower temperature, no, there's limited thermal energy for your particles to move. So that's why hindi sila nakakapunta agad sa mga active site, hindi agad sila nakagawang chemical reaction. So kumbaga, they slow down at lower temperature. Okay? Kasi malamig. Okay? Mababa yung thermal energy. So again, the idea here is that the maximum enzymatic reactions or the maximum rate of enzymatic reactions occur at normal body temperature, which is somewhere around 37 degrees. And obviously, in terms of the pH as well, okay, enzymatic activity tends to peak at neutral pH, okay? So around neutral pH, which is around 6.8, 6.9, okay? So bakit? Kasi our body, sa ating katawan natin, ang pH niyan ay 7 or 6, di ba? So ibig sabihin, our enzymes will work best at those pHs, okay? So tingin nyo, bakit kaya, no? Probably kasi ang daming water. Okay? So the water actually predominates the liquid solutions in our body, di ba? So that's why the pH is similar with the water. Okay? So that's why enzymes should also adapt to that pH. Okay? So dun sila nag-evolve, dun sila lumaki sa pH 7. So over time, um, we found out, no, that enzyme activity is really at its peak kapag normal pH. Okay? Kasi tayo ay gawa sa water mismo. Okay? Basically, yun lang yung mga ganap dyan. Okay, so increasing enzyme concentration increases the rate, obviously. Pag nasa supermarket, mas maraming counters na binuksan, mas mabilis ang checkout, 'di ba? So substrate concentration, as you can see, if you have so many substrates, so many reactants, now overwhelm na yung ating enzymes and they plateau. Okay? Nagsasaturate na sila. Okay? We have this thing here called the turnover number. The turnover number is actually the amount of substrate produced per minute, no, by our enzyme. Okay? So, pag mas malakas yung turnover number, ibig sabihin mas efficient yung ating enzyme. Okay? Pag mas mataas yan. Pag mas mababa turnover number, ibig sabihin, probably yung enzyme hindi siya fit for that purpose. Okay? So again, turnover number is just kung ilan yung na-process niyang substrate per minute, per mole of enzyme. Okay? Then again, optimum temperature. Optimum temperature is around 37 degrees Celsius. Okay? So, when we have fever, ito, it's a nice thing to learn. Okay? So, kapag tayo nilalagnat, why do you think that happens? Okay? Kasi apparently, one of our immune response, no, when we are infected is to kill the protein, no, na nagko-cause ng ating infection. Okay? For example, you have kunwari, ibang coronavirus, no? Even the flu, coronavirus din naman yan. Kunwari, nagka-flu ka or sipon, no? So, minsan, lalagnating ka. Okay, bakit? Kasi gusto niya patayin yung protein na yan. Okay? So, yun yung reason bakit ka nilalagnat, no? So, ako, I highly suggest, kapag kayo nilalagnat, hayaan nyo lang. Ang ginagawa ko, mas lalo ko pinapainit katawan ko pag ako nilalagnat. Nagbabalot ako ng jacket, bahala na pawisan kung pawisan, basta super init na. Tapos kinabukasan, okay na ako. Isang tulog lang. Pagka gising ko umaga, wala na akong sakit. So, kaya pag ako nagkalagnat, usually 2 days lang. Kaya yung first day, hinda-inda ako. Tapos kinagabihan nun, magkukumot ako. Jacket ako na sobrang kapal. Then, papawisan ako buong gabi. Then, kinabukasan, wala na akong sakit. Okay. Recovering na lang. So, ganun. Pero, there are risks as well, no? If your fever is too high, especially kunwari, let's say 40 degrees na yan, okay, it could be detrimental to you as well. Because remember, your body is made of proteins as well, okay? So, if that temperature persists, no, probably, hindi lang yung infection yung mamamatay. Even some of your vital proteins could also die, okay? So that's why kapag may lagnat, okay lang yung mga ilang araw lang, pero pag isang linggo na 'yan, dalhin ka na sa ICU niyan, no? Kasi delikado 'yon, okay? So probably may iba ka pang comorbidities, okay? And then again, in terms of pH, ayan, so remember that last time pH can change the shape of our proteins, so we have to keep that in mind. And the optimum pH is around 6 to 7, okay? However, there are some enzymes that work at lower or higher pH. For example, trypsin, it is in the stomach, so it should be able to survive at those pHs. pepsin pala yun, and then we have the trypsin. Ayun, yun yung mga nasa bile salts, and usually bile salts are basic, okay? Mga basic yung ano na yun. Kaya mataas na pH yung kailangan niya. Okay, so now let's proceed with enzyme inhibition. Okay, so di ba kayo nagtataka, no? Kasi kunwari, bakit kaya di tayo kain ng kain, no? Bakit kailangan lang natin kumain, let's say after 8 hours, after 9 hours, 10 hours, no? Bakit kaya hindi kailangan parati kumakain? Kasi look at the car, no? Yung sasakyan, andar yan, para yan umandar, para yan gumana, kailangan yung fuel tuloy-tuloy. Bakit tayo hindi, no? Ba't yung fuel natin, kain lang tayo ng sangyup, tapos kinabukasan, doon ka na lang uli kakain. Di ba kayo nagtataka? So, ang sagot doon is enzyme inhibition, okay? Because, merong mga nangyayari sa body natin when they produce specific amounts of products, sinasabi na ng body natin, oy, stop na, tumigil ka na sa pagkain. Okay? Pag marami na siya na-produce, let's say, na energy, sasabihin niya, no, stop ka na, antukin ka na, yun, antok ka na. Okay? Pag busog na, di ba? Ako, ganun ako, pag busog na, antok na. Ibig sabihin, stop na ako sa pagkain. So, ganun din yung ating bodies, no? So, our enzymes are somehow inhibited, no? Whenever our body achieves specific amount of the products it needs, okay? So, that's why hindi natin kailangan kumain every now and then, okay? Hindi tayo tulad ng sasakyan na kailangan tuloy-tuloy yung gasolina, okay? So, we can reserve our food in the form of fats, no? And then later on, we can use it again, okay? So, kapag gutom na, kain ka na lang uli, okay? So basically, that's the concept of inhibition. Okay, so the inhibition will tell you when to stop the chemical reaction if you have enough number of products. Okay, so there are two types of inhibition. We have this thing called the irreversible inhibition and the thing called reversible inhibition. So the irreversible inhibition is basically wherein your enzyme is being converted to an inactive form. Irreversible to a sense na hindi na siya mababalik sa dati pa. Okay? And apparently, irreversible inhibitions are hindi siya always bad thing, no? Kasi na-engage nyo, permanently inactivated na yung enzyme. So baka isipin nyo, baka masama yan sa katawan ng tao. But apparently, it's not. Okay? So I googled this paper kanina. Okay? So developing irreversible inhibitors of protein kinase cystinome. Okay? So basically, this paper gives us an idea that this concept can be applied to cancer cells. Okay, because cancer cells are just abnormal cells, no, doing normal enzymatic functions. So sabi dito sa paper na to, or sa isa pang paper na nabasa ko. Okay, so bale sabi dito, we can use that in irreversible inhibition to stop yung production ng ating mga cancer cells, parang huminto na siya. Okay, so yan, so ibig sabihin irreversible inhibitors are not always a bad thing, no, because sometimes they can be used in pharmacological researches, no, in order to develop drugs that could help us eliminate cancer, let's say. Okay, pero in our body, the common type of inhibition we have here is the reversible. Okay, so sa reversible inhibition, pwede ito yung hinto ka muna for now, balik ka na lang after 6 hours, no? So kaya nagugutom agad tayo after 6 hours kunwari. Okay, so yun na yung mga reversible inhibition. So there are two types of reversible inhibition. You have the competitive and the non-competitive. Okay, so pag sinabing competitive, this is where your substrate gets substituted by another substrate. Okay, so for example, look at this picture here. So ito yung original na substrate. Okay, tapos si competitive inhibitor, siya yung pumalit dun sa active site. Okay, so as you can see, your substrate cannot go into the active site anymore due to the presence of the competitive substrate. Okay, so yun yung nangyayari sa competitive inhibition, wherein your inhibitor looks like your substrate so that it can fit on the active site. Okay, So sa active site siya papasok. So as a result, your substrate cannot go into the active site and do catalytic conversion, catalytic reactions. Okay? So yun yung ating mga inhibitor. And actually, umiinom tayo niyan minsan, especially yung mga kamag-anak nating mga may high blood, no? Such as ACE inhibitors, no? So yan. Later, we will take a look into those inhibitors. Okay? So this is an active, competitive inhibitors wherein your molecule attaches to the active site itself, and that causes the enzymatic reaction to stop. Okay. So yan. However, as you can see in this graph, no, ang competitive inhibition, pwede siya mapigilan when you increase your substrate lalo. Okay. So by increasing the original substrate, yun, magiging parehas na yung kanilang speed. So ibig sabihin nun, you can still overturn the competitive inhibition, no? Okay. Basta damihan mo lang yung substrate mo. Okay. So basta marami lang makikipag-agawan sa active site, then yung competitive inhibitor mo, hindi na siya mapapansin ng enzyme. Okay? And then next one is the andito pala yung enzyme regulation na allosteric. So now let's talk about the non-competitive inhibition. Okay? So yung non-competitive inhibition, they mostly fall under this category, the enzyme regulation. Okay? So mostly nito ay mga allosteric inhibition. Okay? So again, not all body processes need to happen always. Okay? There are some just specific times wherein they are needed. Okay? Sa bali ganun. So we need to regulate our enzymatic activity. So paano nire-regulate yan ng body natin? We have 4 mechanisms in doing that. So first one is the feedback inhibition or the feedback control. Okay, and this type of inhibition can be done competitively or non-competitively. Okay, so in competitive, alam natin ang nangyayari dito. For example, this chain will produce the product D. Okay, and it appears that this product D is a competitive inhibitor for A. Okay, so kunwari ito sa line na to, yung D na na-produce natin, it appears na competitive siya kay A. Sa bale, doon na mangyayari yung feedback inhibition. So pagka-produce ni D, may kipag-compete na siya sa kay A. So that means, malilimit na yung production ng D. So hihinto na yung reaction na to. Kasi si D mismo, siya na yung kakabit sa enzyme. So that's the feedback inhibition. Kung ano yung product mo, yun yung pwede mong maging inhibitor. Competitive, let's say. Okay? So yun yung ating feedback control. Okay? So another type of feedback control is the allosteric ones. Okay? So the allosteric one is wherein, di ba alam natin may active site tayong tinatawag wherein your enzyme can go. The allosteric site is the site wherein your different molecule can attach to that. Okay. Ang nangyayari, when a molecule attaches to the allosteric site of your enzyme, again, yung allosteric site, ito yung crevice different to the active site. Okay. So kung ito yung active site, itong crevice na to, we call it the allosteric site. So iba yon dito. So nangyayari when a molecule attaches to our allosteric site, yung shape ng ating active site nagbabago. Okay? So as you can see, ito yung original shape ng ating active site, but when our allosteric site becomes attached with the regulator, nagbabago yung shape niya. Okay? So pag nagbago yung shape niya, hindi na makakapasok yung ating substrate. Okay? So yun yung allosterism. Okay? So instead na yung ating substrate kumabit sa active site, dun siya sa allosteric site. So pag kumabit siya sa allosteric site, magbabago yung shape ng active site so that your substrate, let's say glucose, hindi na siya magagamit. Okay? So one example of this allosterism can be found here. Okay? So this paper. This paper is about cancer pa rin. Pero ayan, pinakita ko sa inyo. So makita natin that Para siyang feedback inhibition din, no? So for example, this is glycolysis. So glucose will be converted to G6P, F6P, F16BP, ayan, so on and so forth. Pagka-produce natin ng pyruvate, syempre may mga ATP din na mapaproduce doon. And apparently, those ATP produced can be used to inhibit this enzyme phosphofructokinase. Okay? So bale mangyayari dito, your ATP can attach to the allosteric site, let's say. So pag dumikit na siya sa allosteric site, hihinto na tong reaction na to. So ibig sabihin, hihinto yung glycolysis. Okay? So that's why kapag tayo, we eat too much na, sabi ng body, oh, stop na. Dami mo nang kinain. So yan. So kasi marami ka ng energy na mapo-form. So stop na niya yung phosphofructokinase. As a result, stop na rin glycolysis. Therefore, yung mga sobrang kinain mo, may i-imbak yon as glycogen. May gimbilbil yun. Okay? And then kapag kunwari gutom ka na, resume uli ng glycolysis. So yung ating bilbil, ma-convert uli into energy. Okay? So ganun yung concept ng enzyme regulation. Allosterism, ibig sabihin na to, a molecule can attach to the allosteric site and therefore the active site will change its shape. Okay? When the active site changes its shape, therefore, yung substrate di na siya pwede dumikit dyan. Okay? So yun. So ito, this diagram here shows that concept, no? Yung tinagalog ko kanila, ginawa lang picture dito. Okay? So for example, this is your enzyme, this is a deactivator. Kapag may activator, the substrate can attach to it. However, when you place your inhibitor to the enzyme, hihinto yung reaction. Okay? So kapag kinabitan mo yan ng inhibitor, hihinto na yung reaction. And then kapag gusto mo na dyan uli gumana, ayan, gana na siya uli. Okay? Another type of enzyme regulation is the proteolytic enzyme. Enzyme mo dyan. So katulad lang to ng sinabi ko about insulin. So this is the insulin or the pre-insulin. So bago yung gumana, 'di ba sabi ko, para gumana yung insulin, kailangan mo putulin 'yan. Okay? So you need to cut this single chain to produce this two chain of molecules, okay, of protein chain, okay? So 'yan yung proteolytic enzymes, okay? So wherein if you want your enzymes to be activated, sometimes you need to cut specific parts of the molecule. So by cutting specific parts of the molecule, it can fold accordingly and therefore it can do enzymatic reactions. So ito yung pinakita ko sa inyo dito kanina. Okay? Oopsie. So this is our pre-insulin, the zymogen, okay? The precursor of our enzyme insulin. Okay? So yan. So papansin mo tuloy yan, pero when you cut this specific portions, mahati siya into two different protein chain. And once our insulin is mature, it can work. Pwede rin ba yan, sir, ibalik dito? Pwede, okay? Kasi apparently, according to my professor, insulin is toxic to the body. So that's why sometimes we have to limit the insulin production. Okay? So we need to return it back to its zymogen form, okay, which is the pre-insulin. Okay? Then sometimes you need to activate it again to lower down the sugar levels. Next one is the covalent modification. So parang ito rin yung PFK kanina, kay the phosphofructokinase na. So ito yung sinasabi ko kanina, para din siya yung kanina sa feedback regulation. So mangyayari, you will change the connections of your enzyme. Okay? In this example, lagyan mo siya ng phosphate. Okay? So pag kumabit yung phosphate sa ating kinase, hihinto na yung ating reaction. Okay? So yun yung covalent modification. However, when you do the reverse, then you can reuse that again. Okay? Kasi again, covalent modification, maglalagay ka ng molecule or magtatanggal ka. Okay? And it follows the concept as well of allosterism. Okay? Then lastly, isoenzyme. So ito naman isoenzyme, these are enzymes that are used in multiple functions. Katulad nga ng sinabi ko, lactate dehydrogenase na can be used to convert lactic acid to pyruvate and vice versa. Okay? Depending kung nasan siya. Okay? So if it is in the muscle, then probably its function is to convert pyruvate to lactic acid. But if it is in the heart, let's say, okay, kunwari nasa heart muscle siya, pag skeletal muscle, siguro ano yan, to lactic acid parate. Kaya tayo nagkakakramp sa muscle. Pero sa puso, ang ginagawa niya siguro is to convert the lactates to pyruvate so that magkaka-energy yung ating heart to pump. Okay? And itong ating H4M4 na yan. Okay, so meaning niyan, these are the lactic dehydrogenases that are found at different organs sa body natin. Okay? So as you can see, these enzymes have different concentration at different organs in the body. Okay? So at liver, karamihan doon is the M4. Okay? And at the heart, karamihan doon is the H4. Okay? So siguro ito yung H4, ito yung um, lactic acid to pyruvate. Itong liver naman, yung M4 dito, probably ito yung pyruvate to lactic acid, something like that. And here are some enzymes that are used in medicine. Wag nyo na siya kabisaduhin. And depending on the amounts of these enzymes, it could actually give you an idea on some diseases underlying sa inyong body. Okay? Wag nyo na kabisaduhin yan. Okay. So as you can see, if you have lactate dehydrogenase, increasing that amount can actually uh, result to heart attack. Okay? Kasi nangyari sa heart attack, kunwari, maraming lactate dehydrogenase sa ating puso. And you know, the purpose of lactate dehydrogenase in the heart is to produce pyruvate. And pyruvate undergoes Krebs cycle. So maraming ATP mapoproduce ka doon. Okay? Krebs cycle and then electron transport chain. So maraming ATP. Ang mangyari, yung puso mo, hindi tuloy-tuloy lang yung tibok niya. Ganun siya ng ganun. So ma-heart attack ka talaga kapag hindi mo na mapigilan yung puso mo. Okay? So yan. So that's why even the amounts of enzyme has to be regulated. Okay? Amylase, ito naman, kapag sobrang amylase daw, probably meron kang pancreatic disease or mumps. Yung mumps ba yung beke? Yung ba yun? Kasi alam ko nagka-beke ako dati. Beke. So kapag marami ka palang amylase, magkaka-beke ka. Kaya pala kapag nung bata ako nagkaganyan na. Okay? Yung swollen yung dito mo. Yung pala yun. Okay? So yan. So, ibig sabihin marami akong amylase. Kaya nagkaka-beke. Tapos alam ko ginagawa ng mga pseudo-scientists nating mga kamag-anak, lalagyan niya ng tina, yung kulay blue. Nabutan niyo ba yun o hindi? Pag nabutan niyo yun, welcome to 1998. Yan. So yan. Mga pseudo-scientists nating kamag-anak. Okay, so anyway, that means even enzyme amounts needs to be regulated because if there's too much enzymes, as you know, maraming tayong products na pwede ma-form. And not all products are actually good, no? For example, if you produce too much energy, tas nasa puso ka, dahil tuloy-tuloy yung tibok ng puso mo, mapapagod yan, heart attack ka. Okay? So, yan. So, enzyme inhibitors used in some medicines. We have the ACE inhibitors or the angiotensin converting enzymes. Okay? So, angiotensin is a hormone that increases blood pressure. Okay? By narrowing the blood vessel. Okay? And then, ang ginagawa ng ating ACE inhibitors is that it prevents the production of angiotensin. Okay? Kasi apparently the ACE, this is an enzyme that converts angiotensinogen. So, kino-convert niya ito into the nabubulol na ako. Angiotensinogen, kino-convert niya into angiotensin. Okay? So, yun yung purpose ng ACE enzymes natin. Okay? Pero yung inhibitors na ng ating ACE enzyme, okay? So, pag pinigil niya yung reaction na to, okay, pag pinigil niya yung enzyme na to, it also prevents the production of angiotensin, okay? So, pag na-prevent na yung formation of angiotensin, then hihinto na yung ating constriction of the blood vessels, okay? So, ganun lang yung nangyayari dito. So, angiotensin converting enzymes. Basically, this uses angiotensinogens into angiotensins. Pag in-inhibit mo yan, you will no longer produce angiotensins. And as a result, hindi ka na ma-high blood. Hindi na magko-constrict yung blood vessels mo. Okay? So, alam ko tinuturok to kapag matindi na talaga yung yung case. Nangyari to kay mama. Okay? Pero if hindi naman masyadong serious, then yun yung mga iniinom na. Okay? And then you have the penicillin. Okay? So, penicillin, although it's not an enzyme, no? It has a structure that can do covalent modification to your enzyme. Okay? So, nangyari dito, basahin natin to. Okay, so penicillin, so it causes covalent modification in your enzyme. Anong enzyme yan? Okay, so walang sinabi kung anong enzyme. So anyway, so basically, this molecule causes covalent modification so that the cell wall of your bacteria will not be produced anymore. So probably it destroys the cell wall. So namamatay yung ating bacteria. Okay? So ayun. And then lastly, ito, transition state analogs. So basically, dagdag info na lang to. Pero hindi naman siya masyadong serious. So pag sinabi natin transition state analogs, no? Transition state is the middle part of your enzymatic reaction. Okay? So if you recall your Gen Chem, okay? Kung kung STEM kayo, if you recall that this part of the free diagram, this is called the transition state, and the transition state is usually analogous with the products. Okay, so transition state analogs are, in a sense, no, they copy yung itsura na magiging product mo. So these are substrates who mimic the transition state of a substrate. Okay. So anong chemical importance nito sa body natin, or significance nito sa body natin? So we have enzymes, okay? So ano yung enzymes? These are antibodies that are the transition state analog of our antibodies, okay? So sila ay immunogen, no? Pero hindi sila pero yung antibody, they are not always immunogen, okay? So again, baka malito. So ang antibody, kasama dyan yung mga immunogens, pero hindi lahat ng antibody ay immunogens. Okay? So kumbaga, immunogen is a subset of antibody. Okay? So ano yung purpose ng enzymes natin? So ang purpose nito is to trigger our body with some chemical reactions without the need of antibodies. Okay? Kasi 'di ba usually when it comes to infection, kailangan malagyan ng mga markers yung yung ating mga cells na kailangan may mga antibodies na may ma-produce, 'di ba? Pero yung ating mga immunogen, they skip that process. Okay? They will just trigger an immune response na. Okay? So yan, saan to ginagamit? So I read a paper kanina lang din. Okay. So apparently immunogens or enzymes are used to inhibit the HIV virus. Okay? So kasi 'di ba nangyari dito, pinapatay ni HIV yung ating mga CD4 cells, no? Yung mga pang-mark natin, 'di ba? So as a result, humihina yung mga antibodies natin. Okay? So ang nangyayari, yung immune gens, no? So since they skip that process, no, yung mga markers na yan, mga antibodies na yan, okay? So they will trigger an immune response kahit walang ganun, okay? So that causes the HIV to be neutralized. Okay? So this is actually a good read, no? Okay. Kasi ayun, yun yung problem with our HIV eh. Okay? Pag nawalan ka ng markers sa body kasi sinisira niya yun, edi maging prone ka na to many diseases. That's why if someone has been infected by HIV, we have this thing called ano tawag doon? Yung mga nagte-take advantage na diseases. May term dun nakalimutan ko lang. Okay. So anyway, so yung mga nagte-take advantage na diseases na yon, such as pneumonia, okay, and tuberculosis, ayun. So, since wala nang antibody para ma-recognize ng immune cells natin na, uy, may sakit ka na, may pneumonia ka na. Okay. So, yun. Humihina talaga yung resistensya ng tao. So, buti na lang may immunogens din naman sa katawan natin. Okay? So, again, immunogens, kahit wala yung antibodies, no, ito-trigger niya yung body mo na, uy, may kalabang kagising. Ayan. So, gagawin niya, pag na-trigger niya yung body na may HIV yan, papatayin niya yung virus. Okay? So, that causes neutralization. So, until now, wala pa rin tayong vaccine against HIV. Although it is still undergoing some researches. May gumaling na daw. Okay? So, that's a good news. Pero, still, matagal pa yung process. No? Siguro nagtataka kayo, no? Bakit ang tagal for HIV? Tapos yung COVID, bilis, no? Apparently kasi, pera yung usapan. Sa COVID, bilyones yung pera na binigay for research. But for HIV, the sad thing with HIV, although we are aware, no, the sad thing is that they are kind of forgotten. Okay, in the Philippines, no, although may mga batas tayo when it comes to HIV awareness, pero yung support wala. Okay, so yan. So I just hope na maging improve yung ating mga researches, especially when it comes to vaccines, because this will not only help the people that are infected, no, or yung mga vulnerable na rin in the future. Okay? So, yan. And again, HIV, hindi yan death sentence. People survive from that disease. So, I hope na as scientists, no, as chemists, as biologists, as doctors, we may have, may we contribute more to knowledge, no, and research tungkol dito sa topic na to. Yan. So that ends my discussion on enzymes. Ang dami kong sinabi today. Pasensya na. Kasi gusto ko talaga tapusin lahat today. So anyway, this topic is just more on memorization lang naman. Okay? So that ends our discussion. Midterm, thank you. And I'll see you again after the midterms period. I hope you learned something today na. And yung natutunan nyo, i-share nyo sa mga kamag-anak nyo. Hiningal ako. Thank you guys. See you again after the midterms period. Bye-bye. Ingat parate. Abay, yung mga paper, basahin nyo. Gaganda yung mga reads dyan. Sige. Bye-bye. Ingat parate.